sure. Okay, great. So right at the end, so all of this is work in progress. And I focus more on the ideas rather than the technical details. But at the end, I'll say things about what I want to do next. And I've also got a page of useful resources, which some of you will have seen before if you came to my talk at the Animal Movement discussion group that James and Rob organized. But it's there if people want to go and look for more resources. Okay, so when we think of animal movement data, we tend to think of stuff like this. So this is a track from a female grey seal that was tagged not far from here. This is the North Sea. This is a southern elephant seal going down from Kerguelen Islands to the Antarctic continent. And these are locations from Weddell seals, and actually I'm working on some of this data at the moment. So we tend to think of tracks, so where animals go in X and Y space. But a lot of the animals that we work on actually have another dimension to their movement, whether it's diving, in air-breathing divers like seals, or the altitudinal dimension in things like raptors, and then also sharks that also vary their depth. So these are the kind of things that I work on, rather than locations in X and Y. But animal tracks have specific characteristics that set them apart from other types of data, in that they have a clear spatial and temporal structure. So you get locations consecutively through time, and also they form these sort of shapes that tell us something about what the animal is doing. And commonly we analyze track data using steps and turning angles if we're going to analyze using a hidden Markov model. And that's because these are measurable quantities that tell us something about the behavior of the animal. If for a given time, for a given unit of observation in time, you have really big step lengths, which you measure step length like this, so the distance between two consecutive locations, it tells you something about how quickly the animal is moving, so displacement. And then the turning angle tells you something about the sort of wiggliness of the movement, how much time an animal is perhaps spending in an area. It doesn't necessarily tell you that, but it can. So the other types of data that are also considered movement data and that I've worked on, I'm going to consider two types. One are sparse location data collected through passive acoustic monitoring. And this is a situation where instead of having the locations recorded at the animal, you've got locations recorded at a fixed point in space, and then the animal comes near it, and then you get a detection of the animal. And this is the situation that I was faced with in the data set that I analyzed while I was still based in Cape Town. This is very badly resolved, but this is an area called False Bay. It's right next to the urban sprawl of Cape Town. It's a bay that's really heavily used by water users, by humans, as well as an array of animals, including white sharks. So the interest in this area is both conserving the sharks, so minimizing human-wildlife interactions, because when there is an interaction between a shark and a human, there's extremely bad press. And sharks are especially white sharks, are actually really endangered. We don't know very much about them. So there was an effort to find out more about the sharks, but also to minimize this human-wildlife interaction. So the approach, and I'm going to talk a lot more about it, I'm just going to introduce these two here briefly. The approach that I took was to cut the time series into regular chunks of time, and actually try and infer the location of the animal in a coarse fashion where we have no observations. Because when an animal is near a receiver, you know that's where it is pretty much, within a radius. But when it's away from a receiver, you don't know where it is. But you can use the sort of sequential pattern of these detections to say something about where the animal might have passed through. And the second example is this vertical movement, just diving behavior. And I've worked on diving behavior in seals for quite some time. So it's actually, in a way, really nice to come back to it now with a few years of thinking about it under my belt. And for air-breathing divers, so these are marine animals that breathe air and dive a lot. The dive is a distinct unit of behavior. So these are 10 dives of a female grey seal over, actually I don't know if it's female. Someone in the audience will, I'm sure. 10 dives over about half an hour. And you can see that the animal is going to some depth, spending some time there, and then coming back up to the surface. And this is enforced by the separation of resources. They have to come to the surface to breathe air, and they choose to go to depth to exploit profitable prey. So you can analyze dive behavior using HMMs because you sort of get around the fact that you need the intervals between observations to be regular by saying dives are distinct units of behavior, and so long as I'm analyzing a time series where the gaps are, if there are gaps, the gaps are random and not systematic, then you can say that these are regularly spaced observations because you've got units, successive units of behavior. So you can extract dive variables or different aspects of diving behavior from these dives and analyze it as the input into an HMM, and that's what I've set out to do. So it's not always that easy or that straightforward. So before we had 10 dives over about half an hour. Now we've got 10 dives over about 3.5 hours. And you can see they're long periods of nothing or maybe shadow dives. And this is because Weddell seals are fundamentally different to grey seals in that they're actually not central place foragers. They don't have a place that they stay and then they go out from it and they come back, because they can haul out constantly on the ice. Grey seals and other seals in the UK and lots of foetid seals tend to be confined to coming back to land. Weddell seals don't have this constraint at all. So they're actually a very different kind of animal. But I wanted to use the high-resolution grey seal data first to trial the method and then move on to the Weddell seals. So this is the sort of two-part vertical behavior that I was talking about. So a lot of the time when we are analyzing animal movement data, we want to know what the animal is doing. In some cases, people actually want to get a better idea of location, and that's actually the situation with the shark data. But often you want to get at sort of behaviors, as it were. And to do this, we can describe different movement modes or states along a time series. And there are generally two drivers or two reasons you might want to do this. One is to understand the movement patterns themselves, see what the modalities are in the data. And the other is to understand the drivers of movement. And you can do this by including covariates into your HMM. And HMMs are something that I've been using over the last few years, but they're by no means the only way to do this. So they are nice in that they're flexible and they're general purpose statistical models for time series, which track data are and movement data are. They are also specifically suitable for cases where you've got several processes giving rise to your observations. So a number of people here won't be at all interested in my next few slides, but for those of you who are and haven't come across hidden Markov models before, they're time series models and they are made up of two processes or levels, classically. So you have the process of the observations, the bits that you can measure about your animal, and then a layer, which is the hidden layer, which is stuff that you can't observe, and this is what we call the state process. So you're assuming a structure whereby you've got an underlying behavior, and you want to, you can't measure it, but you can measure things about it, and you want to make some kind of inferences about that underlying process. So the state process is this bottom bit that I've highlighted in red, and it's serially correlated, which is denoted by these arrows. So one state is linked to the previous state. And it takes on finite values. So say this model has 3 states. So this could be, this t-1 could be a 1, a 2, or a 3. And that stands for each of these. So say we've got a 3-state model. And then every observation we assume is generated by one of these 3 processes, which is associated with component distributions. So the value, let me go and bring up this picture. So the value of S, so the state, so that 1, 2, or 3, chooses which of the component distributions generates the observation that we have. And when I first started using HMM, this picture really confused me because in my mind, we observed this and we wanted to know this, but this tells you about the structure that you're assuming. So you're assuming that this exists, it's something that you haven't observed and can't observe, and it gives rise to these observations that you observe. So yeah, in the beginning I was very confused about the direction of the arrows. And crucially, there are no arrows between these Z values. So you assume that those are actually independent given the underlying state. And because of this dependent structure, even though these 2 things aren't linked, it's likely that the component distribution selected at time t+1 is likely to be the same as the previous one because of this link. So that's how you build in correlation into the time series. And this hidden layer is constructed as a Markov chain, and we assume that it's a sequence of random variables. And the Markov chain bit basically just tells us how the process evolves over time. So there's a one-step dependence, and that's the Markov chain bit, basically. So what we're assuming, given this underlying process is a Markov chain, that given the present, the future does not depend on the past. So if you know the state, you know something about the state, but it means nothing about the previous state. So there's only a one-step dependence in these models, in the state process. So the things that you need to construct a Markov chain are the initial state distribution, which is basically the state that your process is, your system is at the first time step. So if this is a seal and your tag is on and you just tagged it when it's on the beach, then you're pretty sure it's in a hold-out state if you had 3 states. But sometimes you don't know because the tag switches on when the seal goes into the water, or you've got another setup where you don't know what state your animal is likely to be in at the first instance. And so if in my 3-state case, if I'm assuming a 3-state model, then that vector will have 3 numbers in it that will sum up to 1. So in the hold-out situation where you put a tag on the seal, it's lying on the beach, the tag is on, you know that at that first instance you're going to have a probability 1 of it being hauled out, 0 of it being in a deep diving state, 0 of it being in a shallow diving state. So those probabilities have to add to 1. And that's your initial state distribution. The other thing you need are these transition probabilities and these one-step transition probabilities, and this is arguably one of the more interesting things about hidden Markov models. It allows you to learn something about how the process evolves, so what the sort of succession of states is. So this is a small gamma, a lowercase gamma, ij, which we use to describe the transition probabilities. And this is basically telling you what the probability is of transitioning to state j, so the next time step, t plus 1, probability of being in state j given right now I'm in state I. And these help us construct the transition probability matrix, which is a really nice thing to have describing your behavior. So if I've got 3 states, then this will have 3 entries. So this is 1, 1, 1, 2, 1, 3. And these diagonals which tell you something about persistence, because if you interpret this as, like if we read it off here, you can say, what is the probability of being in state 1 at the next time step, given I'm in state 1 now? So actually the probability of staying in state 1. And then here you have gamma 2, 2. So move into state 2 if you were already in state 2, and then 3, 3 again. And these are really interesting when you've got an animal that you want to know what is the persistence of a behavior, for example. How likely is an animal to stay in a foraging state if it enters it? So those are nice in terms of describing your system. So that's all I'm going to say about HMMs. It's extremely brief and definitely incomplete, but just a really quick introduction. So moving on to the first sort of sparse example, I'm going to tell you more about the great white sharks in False Bay. So this is South Africa, the tip of Africa, and False Bay is right next to Cape Town in the southwest corner. And it's a relatively sheltered bay, so it's really popular for swimming and surfing, etc. And you get white sharks here year-round. And this is mainly because of this little speck, which is a Cape Percival colony. And the sharks like to eat the pups in the sort of autumn and winter. So you get lots of sharks aggregating around the island and nearby beaches. And there's been a long-term acoustic monitoring project in place for about 10 years now. And the data that I'm going to present are over a 3-year period between 2003 and 2005. So because of this long-term acoustic monitoring project, we know about sort of how often you see sharks in certain locations where the receivers are. But until now, we haven't known anything about the paths that the sharks follow. And it might be quite interesting in terms of, is there a commute pattern? Are you more likely to see a shark in an adjacent beach in one direction or the other? Because there's a really nice community observer project in Cape Town whereby at the main beaches, there are actually observers sitting above the beaches and watching for sharks. So there's, it would be really nice if they say, okay, I've seen a shark here. With the people on either side, should they be at sort of higher alert or not? So there's an interest in knowing what the patterns of movement are around the bay. So we wanted to get from these static detections to trajectories. And this is work that I did mainly last summer, actually, before I moved here. No, summer before, with David Borchers, Theo Michelot, and also Alison Cock, a collaborator from Cape Town. So the question really was, can we fill in the gaps in where sharks go between the receivers where we don't have any observations about where they are? So this was the setup of the receivers. I'm not sure why the resolution is so bad, but all of these little dots are locations of acoustic receivers that were bottom-mounted hydrophones. And they listen not for natural sounds like vocalizations, but for specific frequencies at which these are the microphones, and these acoustic tags. So this is the tag here, attached to the base of the dorsal fin. So these tags are individually identifiable according to the frequency at which they transmit. So you know which shark is in the vicinity of each receiver. So there were 33 receivers in the water over that 3-year period, and 67 sharks were tagged. So this is the kind of data you see when you name the dots. So you can plot, where you saw the animals, and you've got a time series of locations. But if you plot them, and here at the end, you've got these arrows, so it tells you something about the sequence, how often animals move between receivers. So this is the observations themselves, and we want to know something about the gaps between the observations. So because this data is quite coarse, this bay is about 50 by 50 kilometers, and we're unlikely to be able to do to actually reconstruct locations in any kind of accuracy, we threw a grid over the area. So in this case, you've got the receivers, which tell you something about where you can observe an animal, and what we want to know is what grid cell the animal's in. So in this case, I've got 16 stakes. I've got 1 stake for each of the grid cells. I want to know where my animal was. So these inputs are going to be something about where you saw the animal and how many times you saw it in your time interval, and the state, estimated state is going to be the grid cell. So something to notice about this picture is that some cells contain detectors, but not all of them. So there's some cells in which you can't ever detect a shark. So the detection is only really possible when individuals occupy the same cell as a receiver. And even so, detection is not certain because they're quite big grid cells, and there aren't that many receivers in them, and they're also not evenly distributed. But you can detect, because there are multiple receivers in each grid cell, you can detect individuals with the probability of the number of detectors in the cell, so MI. So you can multiply, you're more likely to see animals in cells with more receivers in them. So you don't only, when you see an animal in a grid cell, you have information about it being there at that time, but you also know it wasn't anywhere else. So I wanted to build that into the model. So we supplied the observations as vectors. So you've got 0. So if in my setup I had a 4x4 grid, this would be 16 long, so 16 stakes. So you're saying that in the top example, I saw an animal twice in cell 3. So that's because it's in the 3rd spot in that vector. So that's how we supplied the information to actually account or include information about not having seen the shark in certain places. And we estimated 2 parameters here, but I'm going to add one later. So the main 2 that we're estimating are the Poisson rate multiplied by how many receivers you had in the cell. So this is the rate at which you expect to encounter sharks at a receiver. And we also wanted the transition probabilities to scale by distance. So you don't want your shark to sort of zip all over the place in your grid cell. You want to include some biological realism and say sharks are more likely to be detected near to where they were last detected, for example. So we've got, these are the transition probabilities that I described before, scan ij, and this is the function of distances, so the distance between cell I and cell j. And what I'm estimating is the standard deviation of that blob. So here my f is the normal density, and we chose that just because it was a straightforward thing, and maybe we'll improve it later. So so at the moment, this simple form basically says you want to know how often you detect a shark in a grid cell, and where it's likely to transition to as a function of distance.
Yeah.
Yep.
Sorry. In your observational vectors, is that a 2 because you detected 2 sharks in a grid cell, or is that the same animal?
That's the same animal twice.
And so how is the time?
So you want to, because we didn't want to be able to detect a shark in 2 different grid cells during the same time interval, we limited it based on how fast we know sharks travel, or something about, we don't know a lot about how fast they travel, but we picked an hour, and the grid cells are about 10 by 10. So say you saw a shark twice in an hour in that grid cell.
Okay.
Yeah.
And do you think that that's, like, seeing a shark twice versus once, is that meaningful in terms of, just because of the way the acoustic detection works, does it mean something if you detect it once versus twice?
It perhaps means if there were lots of receivers in that grid cell, it means it was detected on a different receiver, for example.
Oh, okay.
Yeah. So we kind of classed a distinct visit as, like, it couldn't just be coming back in and out of the detection range of one receiver. So, like, detection had a duration of, okay. I think, like, I can't remember, it was maybe half an hour or an hour. I hope that helps.
Yeah.
Okay, so if we go back to the data and then putting the grid over the top, you might think that, okay, say you're in cell 1, you're more likely to move into cell 2 or 5 and 6. However, and that's just looking at the distances themselves. However, not all distances are equal in environmental space. So this is a picture of the symmetry from the space. So the dark green near the coast is shallow areas, and we know from various previous publications that sharks are more likely to be found near the coast in these shallow areas, particularly at certain times of year. So I wanted to build that into the model as well. And we thought we would scale the transitions, or scale the distance between the grid cells as a function of environmental covariates, and here we chose depth. So to do that, we used something called the least cost distance, and it basically just, it scales the distance according to depth. So in addition to having the spot on rate parameter and the standard deviation of normal density, we also estimated the conductance of the path between pairs of points on the grid. And this is definitely something I need to read more about, because just to understand better what the least cost distance function does and make sure it's doing something biologically meaningful for this application. And just to quickly explain this picture, so this is of a shark, and these numbers are the average depth in the grid cell. So you can see that most of the numbers around the coast, except for on these sort of far south reaches, are quite small. And the scale is telling you something about the cost of moving along those areas. So it's cheaper to move along the coast and near these areas than it is to move out here into the deeper parts of the bay. So I fit the model to one shark to check that it was working, and I fit the model with the Euclidean distances only, and then also scaled by depth. And the AIC seems to think that the least cost distance works a bit better, and I think that just looking at the paths, I think it looks better. So the purple path is the one scaled by depth, and the green one is the one not scaled by depth. And these squares that keep popping up are where the observations were made. So these are the Viterbi decoded states, and we can get a measure of uncertainty. So this is just the most likely state according to the Viterbi algorithm, but you can actually get a probability of being in the state using a slightly different algorithm, which is something I do want to do. So I like the fact that these look more realistic and that the imaginary, well, it's not imaginary, but the shark is moving along the coast. It's a real shark. So that's all I want to say about the shark. So we set out to estimate locations based on acoustic detections. We managed to get something working, and meanwhile it does need some improvements. It is a start in that direction. And but this is a setup in which the states of the HMM actually discrete locations in space rather than functional behaviors or whatever else. And we were able to use habitat to inform the transition probabilities between states, which is always nice. So moving on to something different, that was what I was busy doing until March this year. And then in April, I moved back to St. Andrews to take up a fellowship looking at diving behavior. And I call my project E-News, and it's the effects of ocean properties on the movement ecology of weddell seals. So this is the Antarctic continent, and the ocean that surrounds it is the Southern Ocean. And ocean circulation, well, the Antarctic is important for ocean circulation, and ocean circulation is really important for our climate. Especially in Scotland, we experience that acutely because we have the Gulf Stream, which means that Scotland is much warmer than, at the same latitude on the other side of the Atlantic. And the Antarctic is really important for ocean circulation because a lot of the physical processes that drive that circulation happen here. So these white bits of the Antarctic continental shelf, up to about 1,000 meters, and this is where Antarctic bottom water forms. As ice freezes, the salt is extruded, so it makes this really cold, dense water, because cold, salty water is really dense, and this water sinks. And it sinks kind of downwards, down the shelf, into these really deep bits, and it actually creates a big conveyor that happens sort of in the abyssal plain and then at surface. So that happens in the Antarctic, particularly in the Weddell Sea, which is this bit. So this is the Antarctic Peninsula. This is all the Weddell Sea. This is the tip of South America. And South Africa is sort of directly there. And so my point here was that important things for ocean circulation happen in the Antarctic, and the Southern Ocean is particularly vulnerable to changes because it absorbs heat and carbon faster than other oceans. So important things are happening. Ocean circulation is changing as a result of global climate change, and these parts of the ocean are particularly vulnerable. So that makes it a really interesting place to study. So my PI, Lars, is involved in this project called Orchestra, which is taking advantage of the fact that Weddell seals live in this area year-round, also in the winter, and they dive to depth. So when you want to find out about what's happening in the ocean, you need something that samples it vertically, and that's exactly what Weddell seals do with their diving behavior. So that's a Weddell seal with a tag on its head. These tags are made at the Sea Mammal Research Unit, and they collect information on the temperature and salinity of the water. So Orchestra involves 3 years of 10 tags deployed each year on Weddell seals, and dive data collected simultaneously whether you want to or not, because these tags are built for seals. And in my project, I'm taking advantage of the fact that these data are being collected. Unfortunately, the deployments that were meant to happen this year before my project started, so in February this year, didn't happen due to ship time constraints. So luckily, there is some older data that I'm using as a pilot, but this data will be collected, and if I have a method that is working, then I can extend it to use the new data. So the things I wanted to know about Weddell seals were, what is the form of different types of dives that you see here? And what is the temporal structure of them? So do you see bouts of certain types of dives, or is it all interspersed? And particularly with regards to methodology, you might remember from the picture of the hidden Markov models, you do inference at the level of the state at each observation individually. But there have been some method developments recently that mean that you can do inference over longer timescales and take into account multiple observations and describe a state involving lots of observations. So I'm going to try and implement that kind of model and see if we can actually learn more by doing inference over longer timescales. And then lastly, are certain dives linked to water mass, which links back to the oceanographic project. So if certain water masses in the Antarctic are going to change, and if certain animals rely on them, then there's possible consequences for the population status of those animals. Critically, we get quite sparse data from this area. The tag technology relies on Argos satellites, which can be quite gappy. Sometimes the seals destroy the tags because they're strongly ice associated and they wreck the antennas, and then you don't get any data. So this is the area we want to know about, but the data might not allow us to actually find out what we want to know. So I've borrowed some data from Debbie and the others who work on grey seals to pilot the method on a more high-resolution data set. So these are some seals that were tagged last year on the west coast of Scotland. And you can't see the west coast of Scotland at all, which is unfortunate. But I'm going to use my mouse. So my little mouse thingy is now on Lewis and Harris. It's now on the Isle of Skye. It's now on the Isle of Mull. And these guys are tagged, I think, on Isla. Good, Debbie's nodding. Yeah, so these are 6 seals, and they're doing quite different things based on just their track. But importantly, they have really nice high-resolution diving data. So this is what you saw before, 10 dives, you get 23 points at depth, and these dives are quite short, they're only about sort of 5 minutes, so you're getting a really nice, rich data set there. So I extracted some dive variables from this dive, so I took the maximum dive depth, so the biggest depth in the dive, and I took the standard deviation of depths that are below 80% of maximum dive depth, so the bottom, the deepest 20% of dives, and I took the standard deviation of that, and that tells you something about what the animal, how active the animal is in that depth, in that depth fin, which might tell you something about the dive's function. And then I also took the duration. So all these 3 things go into the data streams. So you can have as many of these as you want in HMMs, which is really great. and I just had these three. And based on these, I want to infer a state. So quite often, and in the past, when I wanted to know about dives, I've thrown away everything else in the record. So you tend to have, you've got a dive and then a surface interval, and this is the period of diving, but sometimes the animal will fall out, sometimes it will just be at the surface in the top layer of the water column, like maybe up to about 1.5 meters, which is the tag threshold for recording a dive. It's not diving, but it's not hauled out, so it's sort of at the surface doing miscellaneous stuff. So I wanted to include haul out and long surfaces at the long intervals at the surface, but without focusing on them. So I don't want to know what kinds of haul outs they do, or what kind of surface intervals they do. I want to know what kind of dives they do, but I can't, in order to treat the data more like a complete time series, I wanted to include them. So I've used momentum for now while I'm exploring the data, and I might keep using it. It's worked out really well for me, but I started out using momentum, and I've provided the haul outs and long surface intervals as known states. So I'm telling the model that I know, don't estimate anything particular about these states. I know that it's haul out, I know it's surface interval. So all of those were given because I wanted to make inferences about dive types. The nice thing is, if you provide known states, you still get transition probabilities between all of the other states. So you're just not finding out about the distributions. So this is a 5-state model, and I wanted to fit a 5-state model because I wanted to have haul outs and surface intervals, so that's 2 states already dealt with, and then I wanted 3 dive states, which I thought in some way would capture the behavior. And there's no stick, I'll just have to point. So in this plot, in this leftmost plot, these are the state-dependent maximum dive densities. So there is a histogram of observations behind here, which fits reasonably well. So you can't see the haul out state, and you can't really see the blue surface state either because they don't have any depth. But what you can see is this light green state, which is these are shallow dives. So obviously there's a lot of density here at the shallow end. Then you've got these transit dives, which are in thick color, which are also relatively short. And then you've got this really big peak because this animal went out to the shelf on the west coast and dived down to about 120 meters for a while. And these are the duration, and the colors represent the same thing. So these are the density of durations for the shallow dives, density of duration for the transit dives, and what I call foraging dives. These aren't necessarily, so the things that I've called foraging dives aren't necessarily that, but it seems likely that they are. And this bit is actually just the tail, because I've cut this off at about 16 minutes, because that's where all of the diving behaviors happen. But then the haul outs, which are in this yellow, and the surface intervals are much, much longer. So I just cut this off and put it in a separate shot. And then you've got the wiggliness as well. The same colors apply. You get sort of increasing wiggliness at the bottom with sort of progression from shallow dives, transit dives to foraging dives. And I can also, so here I've included no information on the locations of the animals, but because I have locations, I can plot these states on a map, which is what I've done here. So you see the green state is the foraging state that I've called. Pink is transit, these yellow triangles are haul out, and every now and again you can see this pale green color. That's super easy to see. But I was pleased that this looks sort of somewhat convincing about what the animal could be doing. We can see that it's coming away from the land and going out to sea to the west, presumably going to find something that's really exciting. And the model thinks that in these areas, the animal's mostly doing foraging dives. And there's that transit behavior to and from those sites. Another interesting thing you can get from this model, and this is just one animal because I was just trialing it out, is you can get time spent in state. And for this animal, that it spent 20% of its time hauled out, 7% in surface intervals, 8% in these shallow rest type dives, and 50% foraging in these deep dives. And then the rest of the time is 15% of transit. So given no location information at all, you can say something about what you think your animal is spending time doing. So I was quite pleased that this was working reasonably well for grey seals, so I moved on to fit it to all of the Weddell seal data because you get far fewer dives. So that seal had 30,000 dives, and this is 30,000 dives from 19 individuals. So you just get a lot less data. So I thought, okay, well, I'll throw all of it. In terms of dive variables, I had to do something slightly different. I could still use maximum dive depth, but I couldn't use standard deviation because these dives only have 4 points at depth rather than 23. So standard deviation is slightly less meaningful in this context. So I used something that I actually, here's my reference, I developed during my PhD, and it's a measure of how wrong these profiles can be. They use a broken stick algorithm the tag collects all of the dive time depth information at a high resolution of every 4 seconds, and then because it can't possibly send all of that back due to constraints of bandwidth, it abstracts the dive profile and ends up with 4 points at depth in the surface points. So because we know exactly what the algorithm is, because it's programmed into the tags here in St. Andrews, you can reverse engineer it and you can't get back to the original trajectory, but you can get an idea of what the error is associated, basically how wide the margin of error is around that profile. So I use that instead as a sort of measure of wiggliness or error or variability at the bottom. And I also included the duration as before. So here the only thing that's different is instead of using standard deviation, I'm using the dive zone index. So here you have a really similar kind of plot. I mean, if you couldn't see what the scale is, and this is 500 meters rather than 100 meters, you'd say this is actually quite similar type of behavior. So it seems like you can generally describe seal behavior, at least posted seal behavior, with these 5 states. What's interesting here is that compared to the previous example, where you had the really deep dives actually also being the most wiggly in terms of standard deviation, here you've got the deep dives being the least wiggly. We are comparing slightly things that aren't exactly alike, so we're not comparing standard deviation with standard deviation. So it might just be an artifact of that. But it might, if it's true, it might be telling us something about how these animals spend time in the bottom, deepest bit of their dive. So that was something that was quite interesting. And this is all of the seals together, without random effects. So remember the picture I showed you where there's like tracks of pink and blue and whatever, and that you can see some structure there. This is what it looks like if you work out the most likely state for weather seals. So they're doing something really, really different, which, even if you knew nothing about seals, you could know that those two pictures look quite different. So if you think, oh, well, that's all of the seals together, and maybe one, all the points are on top of each other, here it is for one seal. So you really, here you've got a little bounce of sort of deeper dives, but in general, it looks nothing like the grey seal tracks. And again, you can work out time spent in states, and here you've got a huge difference. You've got 65% of the time spent hauled out, which you would expect, because weddell seals can haul out all the time. There's always ice available for them to haul out on, unless they've moved out of the ice, which is unusual. And the surface time is also quite a lot higher, 20% of the time. And then the shallow dives are actually really rare. You don't get shallow dives that often. Oh, and MF I've called sort of mid-water foraging dives, and then DF for deep dives. So the bit that is likely to be foraging, although the shallow dives could also be if they're feeding on krill or something sort of near the surface, is actually much smaller, which is interesting because this is a bigger animal. It could potentially spend much more time foraging if, unless there are some physiological constraints that aren't clear. But it's got haul-out substrate available constantly, which is different to grey seals. Okay, so just to wrap up and sort of say, well, okay, what have I learned from particularly thinking about the seal application? What have I learned so far? Have I learned anything new? For grey seals, I think it's quite interesting to be able to compare what you would get from looking at the tracks and trying to infer behavior from tracks, and then actually looking at the depth profiles and trying to figure out what's going on from there. So it's a nice way of sort of coming at the same problem from both sides. And also, it seems like, obviously there's more work that needs to go into whether these states that I've determined are actually anything functional, or if they're just sort of groupings of movement variables and they don't actually correspond to something that we would call, as biologists call, a functional behavior. But it is a way of describing activity budgets without location data. For weddell seals, it's more intuitive that your deeper dives are longer because they're diving to 500 meters. You have to spend time getting to that depth, doing whatever you're going to do at that depth and come back up. So it's sort of less of a surprise in terms of what we saw for the weddell seal data, but it's interesting that the deep dives have less variability in the bottom, so perhaps they're feeding in quite a different way. And once I've included, which actually comes next, included covariates, things like water mass, it'll, I think, be really interesting to see where they're going in the water column, what they're associated with. And there are various things we can try, not only water mass. But that's just the obvious thing that comes to mind, because the water column in the Antarctic is extremely stratified. Think of like oil and water sitting on top of each other. Well, the oil sitting on top. And then the other thing that I want to do, which I will hopefully get to, is to try and look at estimating behavior over longer timescales with these hierarchical HMMs. So this is the bit that is usually, this is the structure that we usually use in HMMs, but you can actually have another layer, which you can think of this as like producing this HMM, and then this one's producing this HMM, and you can have, say you want to know if the animal's spending like a period of time doing really intensive foraging. You would still have all of the different components in that period. You say, okay, the animal's still going to haul out, still going to do surface behavior, they're still going to do shallow dives, but if it's spending a disproportionate amount diving, then you can say something about that being a more intensive period of foraging. So you're adding a bit of hierarchy, and you can choose how long this sort of upper, I think of it as the upper level, but it's actually the lower level of the HMM lasts. And we can use a bit of biological intuition to say, okay, perhaps for a grey seal you might think of something like an hour or 2 hours, but for a weddell seal you might think of a longer time period because they do spend so much more time hauled out. And this was developed last year by Theoni, and some other colleagues at the University of Bielefeld, where lots of HMM development is happening. I'm hoping to visit them and try and talk about this more. So I think that's all. As I say, this is work in progress, so I'd love to talk to anyone who's got ideas. And this is for anyone who wants to know more about HMMs and movements. Thank you.
About the sharks, you said you're interested to see what happens with interaction with humans. Have you thought of, or is it already there in the model? Have you considered having in the model the interaction between humans or even interaction between sharks?
We don't really have any way of measuring that unless a shark actually bites someone. So we know, I mean, these receivers are like 200 meters from the beach. So if it had an antenna, you would be able to see it while you were in the water. So they're near the beach. So the sharks that are coming and being detected at one of those, Musenberg Beach, you know they're near people. There's sharks there every day, all year round. Just some parts of the year there are more sharks than others. So short of, a shark actually making its presence known to a person, there's no way of measuring that interaction. I'm not sure if that's what you're asking.
Yeah, that's what I'm asking. I think you have some information. I mean, you know that during the hot part of the year, there will be people in the beach, and you can see whether this affects the movement of the sharks towards the coast or so on. And then you also observe more than one shark, so you can see whether they move together or move separately.
The problem with this coarse data is that the detector has a range of between half a kilometer and a kilometer radius. So you can say perhaps that two sharks are there at the same time, but whether they're interacting is sort of dangerous territory. You don't know if the one shark is attracted to the other shark or not. You just know they're both there at the same time. So that sort of interaction bit is difficult to do with this data that's quite coarse.
Really great talk, really interesting. I thought that the data you presented on the difference in the time spent foraging between the two seals was really striking. Do you know, sort of based on back of the envelope calculations, whether either of those values is sort of physiologically feasible for those species in terms of, like, especially for the Weddell seals, like, what could they would have to be eating something that was incredibly nutritionally dense to only spend that much time foraging. Is that even possible?
I haven't done any of those calculations, but we think that they feed on ice fish at the bottom some of the time, so those deep dives are likely targeting ice fish, which are very high in fat, and they're quite big. They can be quite big. And the more shallow dives would be sort of smaller fish and like myctophid type things and perhaps krill under the ice. So I think it's possible that they're getting really like high energy prey in the deep dives. But we also know that the time spent hauled out varies across the year. So they spend much more time hauled out in the autumn, so at the end of summer when there's more daylight, and as daylight disappears during the Antarctic winter, they spend much less time hauled out. So that's something that does need to go into the model, which hasn't yet. Yeah, but yeah, that's a really good point. I should try and find that out. Debbie could probably help me with the grey field. But I think so. I'm not sure who was first.
Following on from that, how confident are you that these different types of dives that you've identified with the Weddell fields are that only one of them is a foraging dive?
Yeah, no, it's likely that they're all, in fact. I don't know if the ones that are really, really shallow, sort of in 15 meters of water, I mean, they could be feeding on krill under the ice. So yeah, they're all possibly foraging dives. It's just the deep ones are really more clearly foraging dives. Yeah.
I guess it's obviously a few things to talk about, and that was super interesting. I guess a couple of things. One was about the standard deviation in terms of the wiggliness, and I just wonder how that would be affected if you have a travelling dive. So for like grey seals, for instance, you know, they do these kind of V-shaped dives, and what that would potentially do to the standard deviation, but it's not wiggly, it's just they're going up and down.
Yeah.
The other thing is, I'm a bit worried that my second point may not be very applicable to the weather seals, which might mean that you don't want to do it the greys, but also the importance of the symmetry. So, like, for example, that grey seal going out to the shelf edge is very much travelling and foraging, if it was foraging, but definitely travelling in kind of very shallow water at the start at least. And I just wonder whether you could put in the symmetry, even maybe not, you could do it in a couple of ways. Either you could put it in as kind of percentage of the water column or whatever, but you could also maybe put it in as a covariate in some way, and that would I think be really awesome. And I really hope that weather seals move a lot and have very curious symmetry.
Well, I have discussed this with Lars. So 2 points. The standard deviation thing, we were worried about that and also it's affected by depth. So the deeper you are, the sort of more space you have to vary your depth. But and also perhaps you've got fewer points. But the standard deviation formula includes a sample size, so that's taken into account of. And then I also tried using the coefficient of variation to see if that worked better. And that was just like one big blob. I couldn't see any states in it at all. So I reverted back for now. But I'm open. I mean, I realize it's sort of a problematic or, perhaps not totally acceptable way of doing it. But yeah, Lars and I have discussed the symmetry and proportion of the symmetry used, and also not just the symmetry straight up, but the sort of terrain ruggedness, as it were, the bathymetry ruggedness. Yeah, so we've discussed that. So Lars will give me the data, and whatever data he gives me, so maybe you want to work on him.
I was wondering, the percentages you got for the times dives, is that derived from the equilibrium distribution of the HMR?
Yes, but multiplied by the duration of the behaviour.
Right, okay. That's just wondering about
Okay, yeah.
Thanks.
Tiago, do we have to unmute?
Do we have Tiago?
Does anyone there have any questions? Do we have to unmute?
He should mute himself, I think.
Do I need to press VC mode? Oh. Oh, no, that's just me. Okay. Maybe Tiago can email me questions if he's not here with us now.
Are there any more questions? Okay, well, thank
Okay,
you very much.
thank you. I can just turn this off.